Okay, well, hi everyone. Thanks for joining. My name is Hannelore Waska. I'm in the ICBM MPI Bridging Group of Marine Geochemistry at the University of Oldenburg, and I would like to show you a little review on dissolved organic matter in submarine groundwater discharge and subterranean estuaries. And so I kind of will start with a little recap so that we are all on the same page on DOM geochemistry. Then I will go a little bit into what I think is interesting and important about dissolved organic matter processing and turnover in subterranean estuaries. And then I will go into some results that we've gotten from our model study side and also some perspectives of where we could go next. All right, so let's start with a little introduction. Just the operational definition of dissolved organic matter and dissolved organic carbon. Basically, it's operationally defined as anything that fits through a filter between 0.2 and 0.7 micron pore size, so any aquatic organic matter. And here on this size chart, you can see this blue line showing the filter cut off, and on the left side you can already see that there is a lot of different there are a lot of different compounds that fit into this definition. It could be a molecular building block like glucose, which is just 180 Dalton, but it could be even several thousand Dalton. It could be viruses and colloids even in this fraction. And that means it's a very heterogeneous mixture of organic compounds. Usually it's quantified as dissolved organic carbon, which is the quantity of DOM measured by a TOC analyzer. So we only measure the single atoms of carbon, which means we don't actually know anything about the molecules, and also we don't know anything about the heteroatoms that are attached to it, like hydrogen, oxygen, nitrogen, and sulfur. All right, so why do we study dissolved organic matter? Well, the main reason is that it's food for microbes, and microbes kind of run this world, so we kind of want to find out what they eat. It also keeps trace metals afloat, most prominently iron in the aquatic environment, but it could also immobilize it in the form of organominerals for geologic timescales. And it links different carbon pools, which is important for us. So terrestrial and marine carbon pools by transport through rivers and groundwater. And it has a lifespan of minutes to millennia. So it's very, very broad, which comes from its heterogeneity. And just to put this into context, why do we care about these small molecules? Well, the oceanic TOC inventory is 200-fold higher than that of particulate organic carbon, and that will include any kind of animal and plant that lives there, and is comparable to the atmospheric DRC inventory. So that's a lot of carbon that we need to take into account, and we kind of want to know where it goes. Okay, so how do we quantify it? The most obvious method is the TOC analysis. So we acidify our samples, we combust them, and then we measure the produced carbon dioxide with the infrared gas detector. And that's a very straightforward method, but it's very difficult to calibrate it well and to get a good trueness of the measurements, especially in the low 20 to 50 micromolar range. We can also go and look at the quality of dissolved organic matter, and one of the most popular methods in recent years was to look at the colored or fluorescent component of dissolved organic matter. So we could screen our sample with different wavelengths of light and then look what they emit. And depending on their emission excitation spectra, we could assign specific characteristics to them, or specific provenance or specific turnover. We could go also into a bit more detail, and that's what I'm doing mostly. And we can use electrospray-ionization, Fourier-transform ion cyclotron resonance mass spectrometry, or short ECFDI-CMS. And it is an ultra-high resolution mass spectrometry method, and the electrospray method, or introduction of the sample, ensures that our molecules are intact when we measure them, when we weigh them in a way. So usually to measure these samples, we have to desalinate them and pre-concentrate them, which is shown in the upper left part here in the picture here. You can see this brown color on our solid phase extraction resin. That's organic matter that's been accumulated from our natural water sample. And then it's fed into this mass spectrum. This is a Bruker Solarix in our group, in the lab of our group. On the right side, it has a 15 Tesla magnet. So just for comparison, in Oldenburg, we have the Earth magnetic field has about 50 microtesla. So this is about 300,000 times stronger magnet compared to the Earth's magnetic field. And if you, and this is a huge setup, but most of it is just the cooling in the magnet, and then the analyzer cell is just about this size of a soda can, and there is a cross section on the lower left here where you can see detector plates and excitation plates, and this is where we put our ions, and then they basically start to rotate in this analyzer cell, and they always rotate with the same radii, but they rotate at different speeds. So the smaller molecules, they rotate faster, the bigger ones, they rotate slower. And this rotation is then detected, and it's Fourier-transformed, that's why we have this name, into a frequency plot. And that can then be calibrated into a mass spectrum. So that's how we get our DOM mass spectrum. And if you want to learn more about this, I recommend this great website from which I took all these figures as well, which is here in the bottom of the slide. So now we have our mass spectrum. This is a typical mass spectrum of deep sea dissolved organic matter from Hawaii's Nelha station, about 670 meters depth. The concentration of dissolved organic carbon there is 40 micromole, so it's pretty low. The age, radiocarbon age, is about 5,400 years, so it's been around a while, it's seen a lot. And if you look here, you have on the x-axis the MC ratios, the mass over charge ratio, and most of our samples are ionized, singly ionized, and so that will translate to our mass. So we have a mass range of this sample, in this case, of 200 to 600 Dalton. And on the y-axis, we have the relative signal intensity. And if we zoom into this sample, into one nominal mass, you can see that there is a lot of different masses already on this one nominal mass. So the resolution is really extraordinary. And that means that we can actually use the mass defect that's very specific to each element to assemble our molecular formulas based on, our chemical rules that we have on how organic molecules have to be put together. And when we do this in a very narrow error range, we can usually assign a specific formula to each of these specific masses. And you see that on the bottom left here, those larger peaks are usually CHO compounds, so they contain carbon, hydrogen, and oxygen, but the smaller ones can contain nitrogen and sulfur as well. So we can get two types of information out of this. Yeah, we can get the sum formula that contains the elements. It contains also a stoichiometry, so how much of each element is in there. And we also have the relative intensity of each of these masses. And these two types of informations together provide us with a so-called molecular fingerprint that is very specific for each sample. Good. Now, what do we do with this kind of information? Well, one possibility is to use this so very famous Van Crevelen plot, where we plot the stoichiometry, the elemental ratio of the oxygen to carbon versus the hydrogen to carbon for each of these sum formulas that we assign. And I just show here some examples. So in the bottom, in this kind of brownish box, the aromatic box, we have a black carbon molecule. So that is something that is derived from combustion processes. And on top of that is a polyphenol, so typical for a lignin molecule. And then on the upper left, we have a fatty acid, and we also have a sugar and an amino acid there. And so based on their very specific oxygen to carbon and hydrogen to carbon ratios, they really find their place in this diagram. And so we can derive some structural information based on the location in the diagram. Now this looks very tidy and neat, and we have these different groups and everybody has its place. But remember that I showed you this other mass spectrum earlier. So this is what we actually get from a deep sea sample that's been around a while and has been undergone a lot of processing. And now if we would plot all the molecular formulas that we get in the sample, our Van Crevelen diagram would look like this. Yeah, so it's a little bit crowded. And this is just one sample. Now imagine that you have to measure hundreds of samples and have to make sense of these samples. So this is a bit overwhelming, and how do we get information out of this massive amount of data? And one way to look at it, or the one way that I try to look at it, is a little bit to look at it as like some kind of oracle. Yeah, so the oracle is this huge data cloud. You see that up there, and there's a lot of data in there. And so the answer that you want, or the answer that you get, depends on the question that you ask. So we have our medium here, it's usually R. This is what we use most cases. And in front, you have these poor students trying to think of what questions they want to ask. And so there are several ways how we can get information out of this data cloud. And one possibility is, I don't know, maybe some of you still remember Okay, let me just check. There is, for some reason, a message. Okay. So I don't know if some of you remember, but there used to be these kind of 3D pictures that were just a mess of patterns. And when you look very, very closely, well, you see a dolphin or something. And so the statistical equivalent of looking very closely or squinting and trying to find a pattern would be the principal component analysis or the principal coordinate analysis, right? So you just take the whole molecular fingerprint of all these samples and try to figure out the similarities between them and then plot them in a 2D plot. This is what I'm doing, what I did here. In this case, these are pore water samples from a subterranean estuary on Spiekeroog Island. So beach pore water are all the pore water samples from the beach area there. There is also some pore water from the dune system that's adjacent to the beach and some groundwater from the freshwater lands. The color, blue color is low salinity and red color is high salinity. And so here you can already see very well that different types of samples are separate from each other based on their DOM molecular fingerprint. And in between the beach pore water and the groundwater, you have some symbols which have this light bluish or reddish color. And those are actually pore water samples, brackish pore water samples from the low water line. So these pore water samples have a terrestrial DOM fingerprint that is more closely related to the groundwater, which is several kilometers inland, than to the beach pore water, which is right there, and even the dune pore water. So this is very powerful to see how these DOM samples are related to each other. And it works if the samples come from very, very different environments. But if samples are very similar to each other, it's sometimes not possible to decipher differences only on this principal component or principal coordinate analysis. And so another possibility how we can use this data is to derive indices, molecular indices, which tell us something about how labile is this organic matter. Do bacteria like to eat it? Have they already eaten some of it? And some two examples are the molecular lability boundary, which is here on the left. So it basically operationally defines between an HC ratio, hydrogen to carbon ratio, below 1.5 and above 1.5. And it says the ratio of these two groups will define how labile my sample is. And in a very similar way, we also do that with this degradation index, the IDEC, which is on the right side, except that in this case, we use a very specific bunch of molecular formulas which have been calibrated with radiocarbon age from the Atlantic. So the molecular lability boundary has been used for all kinds of different types of samples, and the IDEC has been used mainly for open ocean marine samples. There are also other ways how we can look at our data. We can correlate the relative abundance of our formulas with DOC concentration or with any other environmental parameter to see how different environmental variables are connected to different types of molecules. Is there a molecular fractionation based on different types of environmental compartments? So this is one possibility. And we can also do group-wise comparisons. So we can say, in this case, on the right side, I compared an infiltration site, the pore water of an infiltration site at the high water line with the pore water of an exfiltration site at the low water line. So after the seawater has circulated through the beach, and we can already see that there is a little less fractionation. So the green samples, they seem to be more labile. This is what comes in. And then these brownish samples, are a little bit older and a bit more degraded. And this is what actually exfiltrates. So we can see some molecular aging of the dissolved organic matter. And we can tease these differences out by using different types of analysis. And then if we want to go complicated again, we can start linking our DOM data. We kind of condense it, boil it down, and then again, make it more complicated by linking it to microbial community data or by environmental and community data, so that we link specific ecological niches to specific types of DOM. Okay, and then just to summarize this, so what we kind of want to do is we want to take our sample and we just kind of pull it apart and we do all our little analysis, and then we try to kind of put it back together again. And the idea is not to necessarily get the original picture, but to get if we want to use that buzzword, to get a mechanistic understanding, so that we really know why the DOM is the way it is, that we can make predictions, how it could be in another time or in another place. All right, then let's go to the submarine groundwater discharge and subterranean estuaries. I'm just showing a few previous studies that are not mine and not comprehensive in any way, but just studies that I thought would be interesting or telling, because they're representative of what we see right now. So what we know about DOM is that on a quantitative level, there is no homogeneous picture. So we see some systems where groundwater is enriched compared to seawater, and some systems where it's depleted. And it seems to be related to exposure and watershed geology, and also the relation between seawater and groundwater end member. And so one really nice paper was by Jackie Webb in 2019, where they did a lot of different systems, and it was really very heterogeneous in the way the effect that SGD has on organic carbon cycling in the coastal system. On a qualitative level, what we often see is that the organic matter from groundwater is often enriched with degraded or alicyclic aromatic compounds, so-called humics, if you will, and depleted in aliphatic compounds. And that's something that we saw here in this Kim and Kim paper from 2017, but also in the Nelson paper, where we see that with low salinity water, we have this association of high abundances of specific F-DOM fluorescent DOM compounds. Okay, we can also see that there are different distribution patterns in the subterranean estuary, and this is what I'm working on mainly right now. I'm trying to figure out what happens with DOM, how is it produced, how is it processed, and what actually ends up in at the low water line or before discharge. And obviously one key paper was the one by Isaac in 2009. I think it was an eye-opener for a lot of us, that you have this kind of estuarine type patterns of dissolved organic carbon, but with entirely different drivers of why it looks that way. And I know there are also very, very focused studies by Natasha and by Carlos on this, how the dissolved organic carbon is processed. And then also there were some papers where they showed that the sources are very important and very heterogeneous. And because I'm a beach investigator person, I'm obviously looking at these beach systems and always think, well, it's just all sand, but it's not. There are a lot of different geological impacts on the dissolved organic matter input. One thing that I also liked is the interaction between particulate organic carbon and dissolved organic carbon, like in this Kim et al. Paper, where they could show that there was kind of a time lag in DOC release based on these organic particles that are incorporated and then moved to, towards the low water line. So in terms of sources, I would say in general, if you want to generalize, we have these marine beach karst system sources, and then we also have these old terrestrial sources, which can also have an impact on the DOC and member concentrations. If we want to look at processing of dissolved organic matter, I would say roughly we can say that we have two kind of main chapters. One is that we have abiotic processing. One possibility is co-precipitation and adsorption with iron minerals. So there we saw in a proof of concept study that we can coagulate specific dissolved organic matter fractions with iron and keep them in the solid phase. We don't know how long, because we have a very dynamic redox cycling. It's possible that it's released again, but it is some sort of transient storage. There is also actually the possibility of Fenton type reactions, so this type of iron cycling, oxidation reduction cycles. There could be some inorganic DOM degradation, even conversion to CO2, but this has not been quantified as far as I know. One other process, and I'm sorry I'm showing hydrothermal fluids now, but Gonzalo Gomez, who did the study, also did some work on pore water samples, but this is just, I just really like this graph because it shows how in our mass spectrum, when we have sulfidic condition, we can actually see that sulfur-containing compounds suddenly show up in this mass spectra. So we actually have a diversification of dissolved organic matter because there is an abiotic sulfurization. And that is something that we've seen on a mechanistic level, so we could reproduce it in the lab, but we've also already seen it in sulfidic pore water systems. So it's out there definitely, and we still don't know exactly how this will affect the stability and the bioavailability of organic matter. But of course, one big part of it is also the biological processing of dissolved organic matter in a subterranean estuary. Again, now from a beach type perspective, unconfined aquifer type perspective. So here is a nice picture of a sand grain from such a beach site in the this kind of greenish color is the quartz, is the mineral, and then these grayish parts this can be some kind of organic matter that's stuck to the sediment. And then if we zoom in here, we can see these little pink dots and sticks here, which are the bacteria that are sitting on the sand grains. If we look closer, we can really see that they are attached to the sand grains. And we have, so in this Proband and R study in 2018, which I really highly recommend, they said that one sand grain can have 10 to 100,000 bacterial cells. And even more impressive is that they looked at the community on each single sand grain, and they found that if you have, I think, 6 to 8 sand grains, you can already reproduce 50% of the microbial community of one system. Now, this was subtidal sands, but I'm pretty sure that beach sands have a similar dynamic. So this is really, really impressive. So it means we have this kind of core community, very opportunistic community, that you need very little to resettle or recolonize a large area as well. And so what do these bacteria do? What do they eat? So one way to look at it is looking at reactive transport. And now from the molecular, from the DOM perspective, we start, of course, with this very tasty stuff. So this is going to be utilized first, you know, acids, sugars, anything that can be eaten up very, very quickly. And then down the line, if that is depleted, we can go to lipids and carbohydrates maybe. And then in the end, we have these structural components. This is in secondary metabolites. So they will eat them if they have to, but maybe not if they have a choice. And then in parallel dazu, because we're in a subterranean estuary, we have this movement through rocks or sands, we will have also depletion of electron acceptors, of course. And now what we have up here with the DOM is what we would call a so-called intrinsic stability. So we assume that whatever the DOM is will define whether bacteria can eat it or not. And this is, of course, modulated also by the electron acceptors. So if you have a lot of oxygen, maybe even the celery will be tasty. But if there is only sulfide left or CO2 left, then you might not even want to pick up these tastier molecules. And then, of course, this is also modulated by temperature, time. And tool is what I call tool is the specific enzyme suit that these microbes have to split up, for example, even structural components. And so this is kind of my Mickey Mouse version of what I think happens along these flow paths in these subterranean estuaries. And so do we see that in nature as well? That's the question. And here is part 3, the case study on Spiekeroog Island. This is kind of the island where we do all our research for the past, where we did all the research in the past 10 years and beyond. And as you can see on this map, Spiekeroog Island is in the northern German part, close to the Dutch border. So here's Oldenburg next to Hamburg and Bremen, relatively speaking, next. And these barrier islands, they kind of border the one sea to the south. Also, the southern part of these islands has usually salt marshes, and the northern part has these exposed sandy beaches. And they have these elongated structures because they still migrate. So these are kind of sand lumps, Holocene, Pleistocene sands with some peat layers and clay layers in between. And they still move. So they still move towards the east. And so that's why it has this kind of comma shape. And on the left side, you can see this kind of structure. I don't know if I can use my mouse here a little bit. Can you see the mouse? Probably. So here in the middle, or on the eastern, western part now, we have the village of Spiekeroog, which is underlined by the freshwater lens. And this freshwater lens has about 40 meters depth. And the island itself has about 6,000 to 7,000 inhabitants, permanent inhabitants, but it can go up to 80,000 people in the summer. So there's a lot of groundwater abstraction. But in general, the freshwater lens is very solid and it's not changing much. We can also see in the eastern part, these little kind of sickle structures. And so we see actually new freshwater lens formation as the island migrates to the east. Yeah. Okay. So on Spiekeroog Island, there was the establishment of a so-called Spiekeroog Coastal Observatory. So this is the island itself and surrounding areas as well. And that includes our beach investigations. We also have a time series station in the Wadden Sea, which follows the water flow between the Wadden Sea and the open North Sea. Spiekeroog is in general a pretty island. It's now part of the Dark Sky Association. So it's a star island. because it has very little light pollution, so you can go there and watch the stars. And then if you go to the northern shore, here on the right side, you can see that it has usually this exposed beach structure. It has so-called runnel ridge structure, or also beach bar trough structure, depending on who you ask amongst the coastal morphologists. And so here on the left side, you have the high water line, and then pore water at low water kind of drains into these tidal channels. And then we have another bar system or ridge here towards the low water line, and then here we have the ocean. So we usually have this kind of bar trough system or runnel ridge system. So there's a lot of research going on the island. There's also infrastructure, there's a lab there. We can stay there overnight, and there is a lot of support from the island to do this kind of research, which is really nice. And just a very generalized picture of what we think the subterranean estuary looks like there. Here is a cross-section of the North Beach. We have the freshwater from the freshwater lands, from the main water body that is discharging in this kind of runnel system that we saw earlier, but also the low water line. And in between, we have two seawater recirculation cells. And then, of course, when we go from the high water line, which is here in the south, towards the low water line here in the north, we have an increase in this reduced species and also silicate and this fluorescent that we also saw earlier in other studies. So we do see this pattern here as well. And this kind of redox zonation, of course, it contracts in the summer because it's hotter, so it will go faster. And then in the winter, it kind of expands a little. And so this kind of two-plume system is a bit odd, because we know from the textbook that we just have this one upper saline plume, and now we suddenly have two. And this is even a very simplified version, because in fact, we probably have a lot more. But for now, for simplification, I think this is a good first approximation. And so we can assume that when the water circulates through these cells, it ages and the dissolved organic matter is pulled off by these bacteria and is used up. And so what happens to it? Can we actually see that in our data? And first, I want to just show you a little bit about the pore water chemistry. It's a very simplified version now, but on the top left, you can see that you have a decrease in pore water salinity going from the high water line to the low water line. And that's because we have a higher hydraulic pressure, so there is more fresh groundwater discharging towards the low water line. And here, the blue arrows are actually the infiltration zones, and then the orange arrows here are the proposed exfiltration zones. So that's the interesting part about this morphology, that whenever you have some kind of dent or some kind of runnel system, this immediately will turn into a discharge system. And if you have any kind of elevated system, it will turn into an infiltration system. But because the morphology changes all the time, also these in- and exfiltration systems change all the time. So that's why I'm saying it's an approximation on this kind of two-plume system for convenience. We can also see that the oxygen concentrations, of course, decrease along depth. And even if we have infiltration, the general pattern is just decrease from high water to low water line. We have an increase in silicate concentrations here along the flow path, which we've seen in many subterranean estuaries before. So that's something that we would expect. And of course, we have an increase in iron, in reduced iron too, here, dissolved iron, because we will start to have this iron reduction becoming rather important in this system. And so in general, we have this kind of typical patterns of environmental parameters that we would expect along a flow path, use up of electron acceptors. Now, if we look at dissolved organic matter, on the top left in orange, we have RDUC concentrations, and we do see that they decrease from the high water line towards here this runnel system, and this is here seawater, by the way. So we have a decrease here before it exfiltrates. And then here at infiltration, it increases again a little, and it decreases a little, but not that much anymore. We do see a consistent increase with FDOM, with this fluorescent DOM. This is actually now measured with a handheld system. Okay, I don't know why, but my window blinds just suddenly had their own life, developed their own kind of life, sorry. Anyway. So FDOM increases is very similar to the silicate increase. It's rather consistent. And this is an FDOM marker, which is called humic-like organic matter. So it is an indicator for humidification or for kind of aging of dissolved organic matter. And then if you remember, I had shown you earlier this EDEX, so this degradation index, and this molecular lability index, and they actually show contrasting patterns. So increased degradation as we move along from the high to the low water line in general and to here, although it goes a little bit down here and it goes up again here. And then the lability also decreases along the flow path. It goes up a little bit here and then it's again a bit higher here. So we do see in general that we have a change in the quantity, so we have decrease, and we also have a change in the quality. So we have an increase in these molecular degradation patterns. And we have different types of degradation patterns, and they all seem to show something rather similar. Just to follow this up, I mentioned earlier that we have this kind of runnel system where we have pore water exfiltrating into these tidal channels, and it's flowing out into the ocean. And we did a time series there too, and we can see that silicate here, the blue color here, this is 2 time series, A to E and then B to F. This was at 2 different times, and we can usually see that silicate with a little time lag increases after low tide in this tidal channel, because it just gets enriched with this pore water. At the same time, DUC decreases. So we have this discharge of lower DUC pore water that's coming out of these beach systems. We also see this a little bit with the degradation index. I don't know what happened here, why it's actually lower. So it seems to be fresher than here, but here we also see that it's more degraded in this system here. So when we have low tide, we have this kind of higher degradation, molecular degradation index in the pore water that's discharging. And we see something similar here. So we have a decrease of the molecular lability boundary here and here during low tide, well, with some shaky patterns. Now, just a very, very brief detour to the abiotic processing to the sediment. As I mentioned earlier, we did one study on coagulation. So we basically just took pore water samples which had a lot of iron in them and let them sit and see what the iron draws, what kind of organic matter is drawn out by the iron and precipitating. And that is a very specific molecular fraction of the soft organic matter. And we see the same when we redissolve those sediments. And so we just did a very, very weak leaching. So this is no Canfield protocol or anything. We just used pH 2 water and did a few leaching cycles. And we can see that in the first leaching round, which is, so on the top, you can see the organic carbon that we released during these different leaching rounds. And we can actually get a lot with just very, very weak acid that's buffered away quickly. So there is a lot of organic carbon that's kind of loosely bound to sediments. But then as soon as we increase here the iron, we get a very specific associated organic carbon fraction here, this organic rich aromatic carbon fraction. So that means that with increasing acid strength and increasing amount of iron that we release from our sediments, we will get a very specific and increasing specificity of organic matter that is being released. Yeah. Okay. Just to summarize. So DOM is a diverse mixture of hundreds of thousands of chemically different compounds. It can be intimidating to work with this kind of data sets, but these new technologies allow in-depth qualitative analysis of DOM in natural waters, and it can tell us a lot about where the DOM comes from and what happens to it. And so in the last 2 decades, I think we showed, or other people also showed, that they produce mostly degraded, FDOM enriched low DOC SGD, mostly through this transit of aged DOM and microbial processing. But we also see that especially when we have this kind of organic rich soils, or when we have some paleo soils, or when we have some peat, that suddenly we can have these pulses of high DOC. And basically the groundwater just brings up old, presumably sequestered organic matter. It brings it up to a very dynamic oxygen-rich system. We don't actually know what happens there. Very likely it's being degraded and being converted to CO2. And then, of course, we have this abiotic processing, mineral interactions and sulfurizations that also occur and that are not very well constrained. Okay, just a little outlook. So what I still think that needs to be done. So I think the global coverage, we can all agree on that there's just areas where we don't know anything about. I'm thinking South America, also Sub-Saharan Africa, with regards to anything or SGD and STE related. One thing that I'm interested in is impact of coastal reconstruction. That is something that is very important here, where we have a lot of beach nourishment to prevent the islands from being blown away. There was a big storm 2 weeks or 1 week ago, and it basically took away the whole beach face of one of the islands nearby. So it's just gone. There is just no more beach there. And what does that do when the beach is gone and then you renourish it? What happens to the biogeochemistry of this subterranean estuary that basically is built up completely new? In our case, on Spiekeroog Island, we will look very closely into this, into the depths. This is a picture down here where I showed, again, this beach face that I showed earlier, but before I only looked here in these very shallow sediments, 1 to 2 meters depths, but now we will actually build these observatories that ideally should go up to 25, maybe even 30 meters depths, we'll see. So we can really see the whole area of this beach face. And that is important because you can see on this right side here, our traditional subterranean estuaries. We have this upper saline plume and we have our freshwater discharge tube, but actually on Spiekeroog, it probably looks more like the lower part because the beach face, as I said, is changing all the time. Morphology is changing all the time. And these small scale 1 to 2 meter changes, they propagate very, very deep into the sediment, 20, 30, maybe even 40 meters deep. So on the left side, this observatory, the colors that you see on top are actually from ERT measurements. So we already know that there is a very high heterogeneity that fits with those models that Gudrun Massmann and Janek Kreskowiak set up. So there is already some proof, observational proof that this happens. And so what does that do with DOM processing or any kind of processing in a subterranean estuary? Yeah, and then obviously we still need more rates. They change very drastically with steps in these systems, very, very high in the top and then very low on the bottom. And then connecting more the solid and the soft phase, I think, is also an important part, which now slowly gains some interest. Okay, now that was a lot of information. I hope you still could take something home with you. I want to thank my group, the funding organization, also all the local stakeholders. The island of Spiekeroog is very supportive. The national park crew is very supportive. And I would like to emphasize how lucky we were that we have had really, really good PhD students. They are here also on the picture: Nele Grünenbaum, Janis Ahrens, Kai Schwalbenberg, Julius Degenhardt, and they really, they were from very, very different expertises, but they supported each other really well. And I think they kind of made this project work too. So that's it. And yeah, thanks again. Any questions?
Thank you, Hannah. Let's thank Hannah about a great presentation. I personally learned a lot and have more things to dig into. So now, guys, if you have any questions, just unmute yourself and directly ask. I see a lot of people in attending. Any questions? Do we have anybody in the chat? If you want to put your question in the chat, you can do that too if you're too shy.
I think there's one raised hand, but I don't know.
Who is the raised hand? Yeah, just unmute yourself, Lino. Just unmute yourself and ask the question. Can they hear me? No, they disappeared. Actually, Hannah, I have a question because we're now trying to dig into this stuff. And we are looking at the ratio.
Hello.
Oh, okay. Let's first let the person ask.
My microphone was disabled. Professor Waska, can you show me the slide number 35? I have a small clarification there.
Okay.
Yeah. Do you think the second upper saline plume you have plotted is due to the beach morphology itself?
Yeah.
I mean, you have seen that we are seeing two curves, right? I mean, one runnel and the berm immediately after that.
I'm sorry? Mm-hmm.
Yeah, there is a study I remember reading. It was about the about the structure of upper saline plume in different morphological settings. Let's say they have done it in 1:6 elevation aspect beaches and 1:12 beaches, gently sloping and steep beaches.
Mm-hmm.
In the simulation based study, they found that the beach morphology itself, and then the kinds of tides they have, like whether it's semi-diurnal or mixed semi-diurnal, it actually affects the structure of the upper saline plume. Do you think this could be that? This is my
Yes, yes, thank you. So there is a lot of factors that influence the circulation through these beach systems, and I'm not a hydrogeologist, but as far as I understand, it has to do also with the beach slope. Beach slope there is relatively shallow, and there are other density-driven circulations that we usually don't have at steep beaches. I think it was called fingering flow. Yannick Kryskowiak did a model there too. So one assumption is that this relatively gentle beach slope, even small changes in the morphology can change the flow pattern on this beach.
good. Okay. Thank you. I liked your illustrations as well on the figures. It's very nice.
Yeah, very nice. Very clear and solved a lot of questions. Thank you.
Glad to hear.
Any other questions?
Hi, can I ask a question?
Absolutely. Go, Amber.
Thanks for a fantastic talk. I was really fascinated by the iron and carbon associations that you were looking at. Can I ask a little bit more about where those samples came from, and if you have any feeling for, what's sort of causing those different associations of the different organic molecules with different leach shapes?
Yeah, so the flocculation study, the coagulation study, we did at the low water line where there was a lot of iron. So we just took some pore water and let it stand at the atmosphere and waited whatever would be withdrawn. And then we kind of measured before and after dissolved organic matter in the dissolved phase. And so we just had a kind of a difference, and that made this fractionation. It's very, and that was a very clear molecular fractionation of this lower part that I showed earlier too. And then we tried to redissolve also the flocculates, and we could refine some of these peaks, although not all of them, because it was not so easy to redissolve the sample. In general, so in the system that I'm working in, we have not a lot of iron, but it's just everywhere. So there's this kind of background iron that's on the minerals that's just everywhere. And so it's not even an iron curtain, because I kind of thought, okay, we have this iron curtain that's just there, and then whatever goes through stops there. But I think we had just the whole beach is just this kind of iron zone where a little bits of organic matter are being picked up. And then, so with this leaching, we really just played around a bit with it, because pH 2 is the pH that we also extract our samples with. So we just started with pH 2, but of course then just everything gets buffered away by the carbonates and everything. So we just kind of added, and then we just took some subsamples from that. But what it tells me, and what we see from terrestrial samples also, is that there is a very specific fraction that very specifically interacts with the iron and also has complexation characteristics. So it's actually complex formation. But then very likely we also have some association that's then on top of that. We have DOM interactions and so on, just sticky stuff. And so this is what I think what we saw in these earlier leachates, in the first leachates. So that's not something that's specifically bound. And so the question is whether microbes even need to get to this part, or whether they can just live off whatever unspecifically stuck there and just eat it and then die, and then somebody else eats them, and so there's a recycling system going on. So yeah, so that's why I think we have these different fractions.
Great, thank you so much. It was really, really fascinating.
So Hannah, related to this, because we are touching on the different fractions, you mentioned about biotic versus abiotic mechanisms of releasing of DOM, correct? Yeah. So have you tried to quantify, I'm sure it depends on the system and other factors such as amount of DOM, probably quality of DOM, salinity perhaps, and the regime of recycling. what is the, like, how much of it is abiotic versus biotic? Can you tell at all?
That's a bit a tricky question. Because the abiotic processing, I think primarily, as far as we know and have quantified, is something that doesn't convert the organic carbon to inorganic carbon. It's just something that puts it in a different compartment or maybe in a different molecular structure, but not necessarily gets rid of it finally. Yeah. Although there have been some strong advocates who said that this kind of Fenton reactions could also produce carbon dioxide. But I mean, in terms of degradation, I think the microbial community just beats everything. I mean, they just sit there on those sand grains. The organic matter comes along and they have a lot of oxygen. They just pick it up. But how long we can kind of store this, for example, on the sand grains, or how the lability for the microbes is affected by sulfurization, these kind of things that I think are some things that we have to consider. So not necessarily as their own process, getting rid of organic matter, but changing it in a way that bacteria may or may not be able to pick it up. That's my personal opinion, though.
Right. I was just thinking about the process of desorption of organic matter, not specifically converting it to CO2, the final product, but just releasing it from abiotic desorption. And I was wondering, what is your opinion of like how much of it is just sometimes even through recirculating with seawater, especially in these recirculating cells, you could, through ionic exchange, release large pools of it, not necessarily through microbial degradation, right? Yeah,
so there is not, as I said, there is not much iron in there, but I think if you have a long flow path, it adds up. And I think there is a lot of organic matter that's stripped out of these systems, yes.
Okay.
But how much, I don't know. I did some calculations back of the envelope because there are some, again, terrestrial, they've done a lot of this work, of course, for soils. Where they calculated ratios of sedimentary iron and then how much it could actually capture, how much organic matter. And I tried some calculation. It could strip a lot of carbon out there, but I should also say that terrestrial organic matter is much richer in these compounds that like to, right? And so for marine systems, it might not be as much, but we do have a terrestrial compound in there, a component in there, right? We have the groundwater there. The question is whether the organic matter that's in the groundwater has already gone through some of these systems, in soils, or whether there is something new added from peat or whatever in the intertidal zone or close to the beach, which then will be deposited on beach sediments or on its flow path. Does that make sense?
Yeah. Okay. Thank you. Guys, any other questions? All right. Should we thank Hannah again? And I will put this presentation in, or Isaac will, I'll just usually send him. Thank you very much, Hannah, again. It was a really, really great talk. Thank you. And thank you everybody for attending.
Yeah, thanks for attending. And I wish you a good day, a good afternoon, a good evening.
Yeah, for us, a new day. Thank you, everybody.
Bye-bye.
Thank you.